Okay. All right. Good day, everyone. Just as we started our discussion, today we are going to be discussing about amino acid catabolism. Remember, while talking about the plate of amino acids in biological system which contribute to the amino acid pool, we made mention of several pathways. One of the pathways we talked about is protein degradation. Protein degradation increases the amino acid pool. Another method is protein synthesis. Protein synthesis increases amino acid synthesis, but specifically synthesis of non-essential amino acid causes an increase in amino acid pool. We also talked about dietary amino acids. When you eat a food that contains protein, and the protein will be digested to increase the amino acid content in biological system. These are basically the major ways or mechanisms or sources through which amino acid pool is being replenished. But there are also an opposite methods or mechanisms or sources through which the amino acid pool is depleted. And we made mention of protein synthesis, where these amino acids are taken for the synthesis of protein. We also made a mention of the synthesis of nitrogen-containing compounds, where the amino acids have been used to synthesize so many nitrogen-containing compounds, like the neurotransmitters we mentioned, like hormones, and so many other nitrogen, especially the nitrogenous base in the case of DNA and RNA molecules. So another method is that when the energy is needed in the system, this amino acid can be converted to energy, or it can be converted to energy source. Energy for the production of ATP. Energy source for production of metabolites, or a storage form of this energy, like glycogen, like triglycerides. Now, before these are synthesized, let's assume that the body does not need the energy. Therefore, it needs a source of energy where it will be kept for future use. So what is done is the amino acid is broken down to some metabolites, and these metabolites will then be used for the synthesis of the glucose in the case of the glycogen synthesis, or the fatty acid in the case of the synthesis of triglycerol for storage. Now, so classically, there are about 6 metabolites or compounds that are produced due to degradation of these amino acids that are involved in energy production, or involved in synthesizing energy molecules that can be used for the synthesis of energy, which are acetyl-CoA, pyruvate, oxaloacetate, fumarate, succinyl-CoA, alpha-ketoglutarate. I've mentioned at least 7 to 6, but acetyl-CoA and acetyl-CoA, acetyl-CoA can be converted to acetyl-CoA, and everything acetyl-CoA can be used for ketone synthesis, or can be used also for energy production, or can be used for the synthesis of fatty acids, which are like a package in form of triglycerol for storage. So on the basis of this, the amino acid carbon skeletons after transaminations are classified as, or the amino acids generally are classified as ketogenic or glucogenic. Ketogenic in the sense that breakdown of product lead to formation of ketone bodies or fatty acids, or in the case of glucogenic, the ones that which the breakdown can lead to formation of the glucose and subsequently synthesis of glycogen. And from the diagram here, we can see Those ones in red classified as glucogenic, and those ones in light blue are classified as ketogenic. Red are being purple, anyone. So the light blue ketogenic, we can see leucine, lysine, phenylalanine, tryptophan, tyrosine, isoleucine, leucine, threonine, tryptophan. And looking at this thing, we can easily say that some amino acids, apart from the glucogenic and glycogenic ketogenic classification, some fall in both. Some are found as glucogenic as well as glycogenic, while you have a very few, just leucine and lysine happen to be exclusively ketogenic amino acids. And I think that's the reason why you will see an amino acid appearing in, for example, let's say tryptophan. I can see tryptophan in 3 different places. You can see tryptophan can be broken down to form acetyl-CoA here, and tryptophan can also produce acetyl-CoA. Also, tryptophan can also be seen here in the production of pyruvate. So, meaning the breakdown of tryptophan can give pyruvate, as well as it can also give acetyl-CoA. So also, you have phenylalanine producing acetyl-CoA, and then you have phenylalanine here forming a fumarate. So in the degradative pathway, we will see how phenylalanine lead to formation of acetyl-CoA and fumarate, and all these are molecules that can be, for acetyl-CoA is ketogenic, while the fumarate is glucogenic, and that classify this amino acid as both ketogenic and glycogenic amino acid. And so also tyrosine can be seen to produce fumarate and acetyl-CoA. When we come here, we have isoleucine producing acetyl-CoA, and then we'll definitely have isoleucine here also forming succinyl-CoA. And we also have leucine forming acetyl-CoA, and we have another leucine here forming acetyl-CoA. So leucine is basically or purely ketogenic because we don't have any other leucine here forming any of these intermediates that can be converted to glucose. So we have threonine. Threonine can be converted to acetyl-CoA, and we also have a threonine here can be converted to pyruvate. So also threonine, we'll also see the breakdown of threonine forming acetyl-CoA and pyruvate alongside our today's discussion. And the last one, tryptophan, which I've already mentioned, which appear also in the formation of acetyl-CoA, as well as in the formation of pyruvate, which we will discuss basically that. So the acetyl-CoA and acetyl-CoA can be interconverted. So acetyl-CoA can be converted to acetyl-CoA can be converted to acetyl-CoA, which will then be used for fatty acid synthesis. Or if the aim is to produce energy, can go into citric acid cycle to produce energy in the citric acid cycle. You will come to know what is a citric acid cycle when you do carbohydrate metabolism. In carbohydrate metabolism, which is the central energy production pathway, you will see how glucose is broken down to produce pyruvate, and the pyruvate is converted to acetyl-CoA, and then acetyl-CoA is then converted to, or is taken into the citric acid cycle to form citrate. And then the cycle continue with the production of ATP and the production of a reducing equivalent. The reducing equivalent will then be used for the synthesis of energy in what we call electron transport chain. So this cycle, as you see, it's citric acid cycle. I know you must have known it somewhere, but this is the central pathway through which energy is produced, because the ATP is also produced here. And in addition to that, there is what we call a reducing equivalent. And this reducing equivalent are also used to produce energy in the process we call electron transport chain, which we will discuss that in carbohydrate metabolism. So that is why if any of these amino acids can be converted to any of the intermediate or citric acid cycle, this intermediate can be used for the synthesis of glucose through what we call gluconeogenesis. And you will also discuss about the gluconeogenesis in the metabolism of carbohydrate. So, like I said, pyruvate come through glycolysis, is converted to acetyl-CoA. You can see the pyruvate here. So if the amino acids like alanine, cysteine, glycine, serine, threonine, and tryptophan are broken down to produce pyruvate, if the aim is to produce energy, the pyruvate can be converted to acetyl-CoA here. And then the acetyl-CoA will now condense with oxaloacetate, enter the cycle by condensing with oxaloacetate to form isocitrate to alpha-ketoglutarate, to succinyl-CoA, succinate, fumarate, malate, oxaloacetate like this, and then condense with another one like this, and the cycle continue. And the cycle continue when you do carbohydrate metabolism. In citric acid cycle, you know what is happening, how energy is produced as this thing is moving along the cycle, how also reducing equivalent are being produced, and how this reducing equivalent are used for the production of energy. So now, if we have the alanine, cysteine, for example, glycine, threonine, being broken down, they can produce acetyl-CoA. That is when they are needed to produce energy. But assuming they are not needed for the production of energy, rather, they are needed for the production of energy source. Since they are glycogenic, they will be used to produce glucose. So what will happen to pyruvate? Pyruvate will undergo another reaction, which is a bypass of glycolysis, because the reaction that converts phosphoenol pyruvate in glycolysis to pyruvate is irreversible reaction. So gluconeogenesis is not a reverse, completely reverse. You can see some reactions are reversible, some reactions are not reversible. I'm sure you must have started glycolysis, and you must have treated all the reaction in glycolysis down to pyruvate. So if you look at the reactions, you will see that some of them are not reversible reactions. So one of them is the conversion of pyruvate, phosphoenol pyruvate to pyruvate. So pyruvate produced cannot be converted directly to phosphoenol pyruvate from that pathway. Therefore, another pathway we call bypass will have to be used. So there are a number of bypass. When you look at the whole glycolysis, when you want to produce glucose from pyruvate or from the product of glycolysis, you discover that you cannot go back through the same route of glycolysis. There are some reactions that need to be bypassed. So you can go along the path, but some specific reaction need to be bypassed. And one of that reaction is the one catalyzed by phosphoenol, conversion of phosphoenol pyruvate to pyruvate. So what happened once we produce the pyruvate now, another enzyme, typically, which is the bypass enzyme in gluconeogenesis that converts pyruvate to oxaloacetate, is called pyruvate carboxylase. So pyruvate carboxylase, we'll talk about the pyruvate carboxylase. What does pyruvate carboxylase do is that it adds a one carbon in form of a carboxyl group, a highly oxidized carbon to pyruvate to form oxaloacetate. So oxaloacetate can now be used for the synthesis of glucose, because oxaloacetate can be converted to phosphoenol pyruvate by another enzyme called phosphoenol pyruvate carboxykinase. We will also see this reaction involving a carboxykinase reaction shortly as an example in one carbon transfer reaction. So also, now when you have the arginine, for example, glutamine, histidine, proline, and glutamine converted to alpha-ketoglutarate, that alpha-ketoglutarate along or inside the citric acid cycle can be converted to succinyl-CoA, can be converted to succinate, fumarate, to malate, to oxaloacetate. Then the oxaloacetate will now go through that pathway of phosphoenol pyruvate carboxykinase to produce the phosphoenol pyruvate, which phosphoenol pyruvate can go backward off in the glycolytic pathway until where the other bypass also come. So when you do gluconeogenesis, you will see all these bypass I've been talking about to produce glucose. So also isoleucine methionine turn into succinyl-CoA. So also the ones that produce fumarate can be converted to oxaloacetate. The one produced succinyl-CoA can be converted to oxaloacetate. So also ketoglutarate to oxaloacetate, finally to glucose. And then the glucose can be converted to glycogen. The same thing also, the ones that are broken down to form acetyl-CoA can be converted to acetyl-CoA by, we talked about that because there are some reactions involved in that conversion. So acetyl-CoA can now be used to produce a fatty acid by an enzyme fatty acid synthase. So the fatty acid can now form the triacylglycerol, which can also be stored. So this is basically the summary of what we will be discussing in this. Please, you should take your time and listen to those things so that you will not have a difficulty in explanation in the class. So also, like we talked about some reactions in amino acid metabolism in the previous classes. So this class will also talk about some reactions that are very important for what we'll be discussing. One of them is transamination reaction, which we already talked about. And we say transamination reaction is basically catalyzed by a group of enzymes or an enzyme called transaminase. And we said each amino acid has a specific transaminase. And we talked about the mechanism of transamination reaction involving pyridoxine phosphate. And how does the conversion takes place. Specifically, we talked about that, where we made mention of ping-pong mechanism, if you can remember. And the other one, we have also said the aim of this reaction is to get rid or to convert or to transfer the amino group from a particular amino acid to mostly, in most cases, to alpha-ketoglutarate forming glutamate. Then the other one is one-carbon reaction. What is the one-carbon reaction? The one-carbon reaction is simply the transfer of one carbon molecule. Transfer of one carbon molecule from one molecule to another. One carbon from one molecule to another. So it depends. This one carbon, is it in oxidized form or in reduced form? So I can tell you, it can be in both. Depending on what it is doing the transfer. There are quite a number of molecules that involve in transfer of one carbon. We have number one, we have a biotin. Number two, we have S-adenosylmethionine. Number three, we have folate in form of tetrahydro or folate. So biotin is involved in the transfer of highly oxidized or oxidized carbon, mostly in form of carboxyl carbon. So we have S-adenosylmethionine. S-adenosylmethionine also is an opposite of biotin. Instead of oxidized, S-adenosylmethionine transfer a reduced carbon in form of methyl group, CH3, while the oxidized, I said, in form of carboxyl, COO, the COAH. Okay, now folate in form of tetrahydrofolate can can transfer carbon in different oxidation states. Highly oxidized and less oxidized or highly reduced, because it can transfer methyl group, this which we shade as adenosylmethionine group. It can also transfer methylene group, which is more or less slightly oxidized. And then we have a methynyl, formyl, and formimide, which you will see in some of these reactions involving the amino acid catabolism. Now we can see the structures of this by my right. We can see the structure of these molecules. We can see that of biotin here, and we will see a classical example where biotin is involved in a carbon transfer reaction. We also have this as S-adenosylmethionine. We'll see how the S-adenosylmethionine is formed and how does the S-adenosylmethionine is involved is involved in the transfer of the methyl group. And here we have the tetrahydrofolate. Tetrahydrofolate is made up of a 3 group comprising of 6-methylserine. We have 3 amino benzoate and a glutamate. How is this tetrahydrofolate is involved in the transfer of a one carbon? We can see the role played by this N10 and N5 because mostly depending on the oxidation state of the carbon, it can be attached to N5 or it can be attached to N10 or it can be attached to both. That is why sometimes you will hear maybe the prosthetic group or when a reaction involving the use of tetrahydrofolate says maybe N5, N10 tetrahydrofolate was used or N5 tetrahydrofolate was used. So usually when it is mentioned, then the group that was attached is mentioned before tetrahydrofolate. For example, usually the highly reduced methyl is attached to N5. You will hear N5 methyl tetrahydrofolate, meaning to say the methyl group is attached to N5 and it is the methyl group that is being transferred to the molecule that is involved in the 1 carbon transfer reaction. So also you will definitely see how is that being utilized. Now talking about the role of biotin, I have here some examples of how biotin is involved in carboxylation reaction. I have a typical example of pyruvate carboxylase, which I just mentioned as one of the way through which the pyruvate form in this process you talked about during maybe combustion of the carbon skeleton or keto acid produced from amino acids, and they want to be converted to glucose or to glycogen. Then the pyruvate can be carboxylated, where a 1 carbon a carboxyl group is attached to pyruvate to form an oxaloacetate. And we know that oxaloacetate is an intermediate of citric acid cycle or a compound of citric acid cycle or compound involved in citric acid cycle. The compound involved in citric acid cycle, we call them citric acid cycle intermediate. So I will be mentioning this word so that since you've not done carbohydrate metabolism, you will not find it misleading. So you have this pyruvate being carboxylated to oxaloacetates, and what is added is the carboxyl group. The biotin is involved in the transfer of the carboxyl group. And we can see here from the, forget about this BCCP. From the original structure, I think you can see it is not there. So let's forget about it. I was just trying to find a picture that can help me explain this, and I couldn't get any better picture. So I got this. But the aim here is to tell you how is the carboxyl group first is attached to biotin, and then the biotin is involved in the transfer of the carboxyl group. Here you have an enzyme called the biotin carboxylase. Now we'll take off the carboxyl group and attach it to biotin. And we can see the N, the nitrogenous group here. When this biotin carboxylase uses an energy in form of ATP to attach a carboxyl group to this, then you have a carboxylated biotin here. Now it is the carboxylated biotin that will transfer the carboxyl group to the substrate. In this case, the substrate is acetyl-CoA and leads to formation of malonyl-CoA. Typical of this reaction, where acetyl-CoA carboxylated to form malonyl-CoA by an enzyme acetyl-CoA carboxylase. So within the acetyl-CoA carboxylase, because it requires biotin, there is an active site of this enzyme that performs this biotin carboxylase reaction. So when this carboxyl group comes into the active site of acetyl-CoA carboxylase, the carboxyl group is first transferred to biotin to form carboxylated biotin, as we can see here, by a particular active site of the enzyme. Remember, you can have an enzyme having more than one active site. So this enzyme will, this active site will convert biotin to carboxylated biotin. And the carboxylated biotin will now transfer the carboxyl group to acetyl-CoA, leading to formation of malonyl-CoA. So another active site of the enzyme that perform carboxyl transferase will now transfer the carboxyl group from carboxylated biotin to malonyl-CoA, leading to formation of malonyl-CoA. And remember, this reaction requires energy in form of ATP. And where does the energy being used is the reaction between the biotin and the carboxyl group, then which lead to formation of the carboxylated biotin. I hope that explains all the other reactions here. So the same thing happened also in the case of pyruvate carboxylase, which lead to formation of oxaloacetate. And also another reaction that involves that is conversion of propionyl-CoA to dimethyl malonyl-CoA. We will see how some amino acids were even converted to propionyl-CoA, and then the propionyl-CoA is converted to dimethyl malonyl-CoA. Dimethyl malonyl-CoA is also converted to L-methyl malonyl-CoA. The L-methyl malonyl-CoA is finally converted to succinyl-CoA. So just remember the amino acids that we state are broken down to form succinyl-CoA. So these amino acids pass through propionyl-CoA. And then this particular enzyme involves carboxylation, the propionyl-CoA carboxylase to produce dimethyl malonyl-CoA, which we will see the reaction in the subsequent explanation. So also, here we have S-adenosyl methionine. So first of all, before we see how the S-adenosyl methionine performs its function of one carbon transfer, how does the L-adenosyl methionine first form? S-adenosyl methionine is formed from methionine, where methionine reacts with ATP. Methionine reacts with ATP to give S-adenosyl methionine, and this reaction is catalyzed by methionine adenosyl transferase. Now you can see ATP here, the structure of ATP here, only here. You know what is ATP? You have an adenosine, and adenosine is adenine attached to the ribose sugar. Sorry, attached to the ribose sugar. Okay, it's attached to the ribose sugar, and then you have the phosphate group attached. That's what makes it to be a nucleotide. So what happens is in the reaction, this phosphoester bond is being broken down, leading to formation of a bond between the methionine and this carbon, which is the carbon 5 of the ribose sugar, leading to formation of S-adenosyl methionine, meaning adenosyl group is attached to the sulfur of methionine. Now you can see this methyl group of methionine. Now it can easily be removed. So in any reaction involving this S-adenosyl methionine, which indicates methylation reactions, meaning the transfer of one carbon in form of a methyl carbon. So this methyl group is being attached to whatever reaction it is involved. For example, now you have a methylation reaction involving a different type of methyl transferases. You have this R that need to be methylated, where a methyl group need to be added. So this methyl transferase will now pick the methyl group from this enzyme, from this S-adenosyl methionine, and attach it to the R group, forming a methylated R. And then what you have S-adenosyl homocysteine, which with the removed methyl group. Now, S-adenosyl methyl homocysteine can be undergo hydrolysis by an enzyme hydrolase to produce the adenosine and the homocysteine. Now, the homocysteine need to be converted to methionine for another S-adenosyl methionine to be formed, and the reaction continues like this. How is homocysteine produced? I don't know, you will definitely talk about synthesis of methionine, and probably we will talk about this process involving methionine synthesis, where homocysteine will be methylated. So another methyl group is attached to homocysteine. But in this case, it's not some that are contributed to the methyl group. Rather, it's tetrahydrofolate. So you have, like I said, tetrahydrofolate can also transfer or involve in the transfer of a reduced methyl group. So this tetrahydrofolate called N5-methyl tetrahydrofolate contains this as N5. Remember, I show you the N5. This is not the complete structure, but just to show us where the methyl group is attached. It is attached to N5. Now we have N5-methyl tetrahydrofolate. So N5-methyl tetrahydrofolate now donate the methyl group to homocysteine, catalyzed by methionine synthesis to produce methionine. Then you have the tetrahydrofolate with the methyl group being removed. And this reaction required coenzyme B12. I know when you come to amino acid synthesis, you will definitely talk about maybe the role of this coenzyme B12. But for the sake of this explanation, it's just for you to know how the methylation or one carbon transfer takes place. And I've already explained how tetrahydrofolate is involved, but we can see the most reduced, where I said N5-methyl, and we have seen example of that. And we have here the partially oxidized, where we have this hydroxymethyl group in form attached to N5 N10 methylene tetrahydrofolate. So when it is involved, usually it's involved in the transfer of methylene group. Usually a halfly oxidized methyl group in form of hydroxymethyl group. So in that, you also have a formyl group, more or less like a carbonyl carbon, like an aldehyde carbon called a formyl. So it's also attached to N10. So it forms N10 formyl tetrahydrofolate. So in any reaction where this transfer of carbon in this oxidized form is involved, then N10 formyl tetrahydrofolate is involved. You also have formimino, where you have this carbon then are double bonded to nitrogen. That is why the formimino comes from. Then you have it attached to N5. You have N5 formimino tetrahydrofolate, whenever this group is involved in the transfer. So you can see this one involves the transfer of carbon, at the same time also nitrogen. So this is basically one carbon transfer. So another reaction we need to talk about is, this one is not a one carbon transfer. Another prosthetic group called tetrahydro biotin. Remember, we talked about the biotin. They are all prosthetic group or cofactor found in the active site of an enzyme and involved in the reaction. So we have, we talked about S-adenosine methionine. We talked about tetrahydrofolate. We talked about biotin. Now, another one is tetrahydrobiotin. Tetrahydrobiotin does not involve in one carbon transfer. Rather, it is involved in oxidation reduction reaction. We talked about a reaction involving oxidation of phenylalanine to form tyrosine by an enzyme called phenylalanine hydroxylase. How does this reaction take place? Or how does this oxidation take place? The oxidation basically involves a reducing equivalent, NADH plus H. It serves as the source of proton. Yes, somebody will say it's an oxidation reaction, but you are talking about an electron donor or proton donor, which proton donor is basically involved in a reduction reaction. So yes, it's basically involved. Why? Because phenylalanine hydroxylase is a type of mixed function oxidase enzymes, which I mentioned in the class where I was discussing in the previous classes. And we said the mixed function oxidase is involved in oxidation reduction reaction, basically involving the oxygen as a final electron acceptor. So the electron donated here actually goes to the oxygen, and that leads to conversion of oxygen to water. So you have the proton, first of all, donated to dihydrobiopterin, by an enzyme called dihydrobiopterin reductase that converts to dihydrobiopterin to tetrahydrobiopterin. The tetrahydrobiopterin now is involved in the transfer of these protons or electrons to water, to one molecule of oxygen forming water, and then another molecule of oxygen will be used to oxidize the phenylalanine to form tyrosine. We will also see how does this reaction takes place in the breakdown of phenylalanine and the tyrosine pathway and what role it plays. Okay, now starting with the first set of degradation pathway. We have mentioned or seen some set of amino acids that are involved in the formation of pyruvate, which include alanine, tryptophan, cysteine, serine, glycine, and threonine. How are these amino acids converted to pyruvate? Now we will see. I've already given an example in the class or in the previous class where alanine by alanine aminotransferase is converted to pyruvate with the amino group being transferred to alpha ketoglutarate. And we've already mentioned that this reaction involves a transamination reaction or transaminase, which we say it's involved the role of pyridoxal phosphate. So we now know that alanine can be converted to pyruvate. Another thing we need to also see is with regard to tryptophan. Tryptophan is an amino acid that is basically, when you look at the structure, you will see a structure of alanine attached to the indole group. The indole group is nothing but an aromatic ring and a pyrrole ring. So through about four-step reaction, I wanted to give all the step-wise reaction, how this thing is done. But I discovered that these very confusing steps in the metabolism of tryptophan. So I decided just to use this that was used by Leminga, which will make things easy for you. So through four-step reaction, tryptophan lead to the release of alanine, this group, which is basically alanine. So through the four-step reaction, we have alanine release. And that alanine, I've shown you how alanine can be converted to pyruvate. Yes, of course, tryptophan can also be converted to acetoacetyl-CoA. Then we'll see how tryptophan is converted to acetoacetyl-CoA, which is finally converted to acetyl-CoA. So those amino acids that will be converted to acetyl-CoA, tryptophan will definitely be part of them, and we'll see how this tryptophan is converted to acetoacetyl-CoA and finally to acetyl-CoA. Now we've seen tryptophan, a part of tryptophan converted to pyruvate, and alanine converted to pyruvate, or for alanine is formed from tryptophan, and then alanine converted to pyruvate. Now the next one is cysteine, which we've already known also. While talking about non-oxidative deamination, we talked about the reaction of cysteine by amino acid disulfide, where we said it's released the hydrogen sulfide as well as the amino group, leading to formation of pyruvate. Another reaction we have also seen while talking about also non-oxidative deamination is the reaction of amino acid dehydratase, specifically serine dehydratase, where we said serine release water or undergo hydration reaction to release water. And then also as another reaction simultaneously that leads to the removal of the amino group, which is the deamination reaction to form a pyruvate. So basically from this, we can see the four amino acids, tryptophan, so completely serine is converted to pyruvate, cysteine completely to pyruvate, alanine completely to pyruvate. while a portion of tryptophan is converted to alanine and alanine finally converted to pyruvate. Now we are left with glycine, threonine, and then which other one? Threonine and glycine. Okay, let's talk about threonine and glycine. How threonine and glycine are converted to pyruvate. So let's talk about threonine. Threonine is just like serine. It's an amino acid that contains this hydroxyl group, but it has in addition a methyl group attached to it. So an enzyme called threonine dehydrogenase, which uses a reducing equivalent NAD, will oxidize threonine by collecting two protons from threonine. And once two protons from threonine is being removed, this particular proton attached to, this is alpha carbon, this is a beta carbon, attached to the beta carbon is being removed. Once they are removed, this leads to formation of a carbonyl carbon. And formation of carbonyl carbon will lead to a compound or formation of a compound we call 2-amino-3 ketobutyrate. These 2-amino-3 ketobutyrate will be ligated or will be attached to coenzyme by an enzyme called 2-amino-3 ketobutyrate CoA ligase. This CoA is coenzyme E. It's coenzyme E, and it is a way of, for example, activation. So any reaction where this is attached is mostly an activation reaction, making the molecule a high energy molecule. So in this case, it's facilitated the breakage of this group because this CoA is attached here. So attaching here, because we know carbon cannot take more than 4. The outermost shell of carbon cannot take more than 4. Then that facilitates the breakage of this bond. So and formation of a new bond containing the coenzyme. Thereby, this group with coenzyme A is what we call acetyl-CoA. So acetyl-CoA is formed. If you can remember, in threonine, we talked about threonine producing acetyl-CoA apart from pyruvate. So while discussing those amino acids that give rise to acetyl-CoA, we don't need to mention threonine, or we don't need to talk about how threonine is converted, unless if there is a different pathway where it is converted to acetyl-CoA. Otherwise, we don't need to. So this cleavage of this to form acetyl-CoA, and this group, when you look at it, this group, Alan, is now another amino acid we call glycine. Remember, glycine is the only amino acid that have a hydrogen as a side chain. So this side chain is hydrogen in this. So it forms glycine. So this is the glycine. So how is the glycine now converted to pyruvate? There are 3 different pathways through which glycine is broken down, but only one out of these 3 leads to formation of pyruvate. The most common and the predominant one is the one that does not lead to pyruvate, the one that leads to complete breakdown to form carbon dioxide and ammonium, which is the one catalyzed by glycine synthase, also called the glycine cleavage enzyme. So this is more or less an oxidation of glycine to form a reducing equivalent. And then, when you look at the structure, so when you look at the structure now, glycine synthase, which catalyzes a reversible reaction, or glycine cleavage enzyme, remove this amino group, which we can see it here, and this carboxyl group, which we can see it here. And this group, which is the methylene group, is now transferred to tetrahydrofolate to form N5N10 methylene tetrahydrofolate. So it will serve as a source of carbon in any carbon transfer reaction. So you can see this reaction give rise to this. It uses tetrahydrofolate to convert it to N5N10. So with this thing you are seeing, you can see tetrahydrofolate here. When it goes, so it is involved in this reaction, lead to formation of N5N10 tetrahydrofolate. So in this reaction, which is another pathway of glycine degradation, in that reaction, it uses N5N10 methylene tetrahydrofolate, where N5N10 methylene tetrahydrofolate like attach or contribute or give a methyl hydroxymethyl group to glycine, leading to formation of serine. So this is two different pathways. Now just imagine a pathway where glycine is converted to carbon dioxide ammonium, and then a reducing equivalent here we call NADH plus H, and then it also converts tetrahydropolate to N5N10 methylene tetrahydropolate. That is a separate pathway. Now another pathway, you have glycine. Now glycine in that pathway is converted by an enzyme called serine hydroxymethyl transferase. What happened here? You have a methyl group attached to hydroxyl group. We call it hydroxymethyl. So you have this N5N10 tetrahydropolate containing this methylene group. And this methylene group is the N5N10, is the partially oxidized carbon, which is in form of hydroxymethane. So this hydroxymethyl group is now transferred to glycine to form serine. And you can see from the reaction, you can see where the hydroxymethyl group is being attached to. When you look at this compound without the hydroxymethyl that is newly attached, you can see you have the glycine, just as where you have here. So attaching of this hydroxymethyl group gives what we call serine. And we have already explained how this serine can be converted to pyruvate by serine dehydratase. Exactly. So this is the reaction that leads to formation of pyruvate, while the one by glycine cleavage enzyme does not lead to pyruvate. But this is the most common one. But there is another also that involves amino acid oxidase. So basically, because this amino acid, this glycine can be degraded by amino acid oxidase to produce what we call glyoxylate. You will know what is the glyoxylate, you know what we call glyoxylate cycle when you go into carbohydrate metabolism. So, like we have said, in amino acid oxidase is a monooxygenase or mixed function oxidase that use an oxygen to produce a water, or sometimes we say it even produce hydrogen peroxide, which later converts to water. So, amino acid oxidase deaminate glycine and at the same time produce water. They form glyoxylate. Now, glyoxylate is being reduced to form oxalic acid by an enzyme which is an isoform of lactic dehydrogenase to form an oxalate. And this oxalate can react with calcium in the body to form calcium oxalate crystal. And this calcium oxalate crystal, when in kidney, they can accumulate to form or build up to what we call a kidney stone. So it's responsible for about 75% cases of kidney stone. We've seen how dangerous this reaction is. Okay, now we have seen all the enzymes, or all the 5 enzymes, how these 5 enzymes are converted to pyruvate. And we've known that pyruvate, after being produced, can be converted to oxaloacetate by pyruvate carboxykinase, which we've seen in the first slide of this reaction. And then can find its way, the oxaloacetate can find its way through phosphoenol pyruvate carboxykinase to phosphoenol pyruvate, and then can be used for the production of glucose. So the next one now is formation of acetyl-CoA. Now, before we talked about formation of acetyl-CoA, we talked about nitrogen-containing compounds. For example, in the degradation of tryptophan, we said tryptophan is degraded to form phenylalanine, and a portion of it will be converted to acetyl-CoA. But of course, also, the tryptophan can be converted or can be degraded to form some compounds that are needed in the body, like nicotinate. Nicotinate is the precursor of N-adenosyl, NAD and NADP, nicotinamide adenine dinucleotide, and nicotinamide adenine dinucleotide phosphate, which we call a reducing equivalent. Usually, they are used in oxidation reduction reaction. When you do in the carbohydrate metabolism, you will be told that NAD mostly, this is not in all the cases, mostly NAD is used in degradation pathway, in the breakage, in catabolism, where the NAD is converted to NA, or can be used to produce NADH plus H, and the NADH plus H, it is used for energy production and electron transport chain. While the NADP usually serve as the sources of electron or a proton in most of the oxidative and reductive reaction. So they serve as a source. If a reaction need to be a particular metabolite need to be oxidized, then NADP can come and can collect the proton, and then the molecule remain oxidized. Or if it needs to be reduced, the NADPH plus H will come under the proton, and it will remain in the reduced form. So this is basically in the reductive pathway, which more or less of biosynthetic pathway, while NAD is more or less in catabolic pathway. So tryptophan can be used to produce this molecule. Tryptophan can also be used to produce another neurotransmitter called serotonin. It can also be used to produce indole acetic acid, which is a plant growth hormone. So just also like phenylalanine and tyrosine can also be used to produce dopamine. The dopamine is also another neurotransmitter, and phenylalanine can also, tyrosine can also be used to produce hormones like adrenal hormones like epinephrine and norepinephrine. And they can also, or they've been used also to produce a skin pigment and hair pigment called melanin, which is responsible for being white skin or dark skin or white hair or dark hair. So this is basically the degradative pathway leading to formation of this. In addition to this, but remember, this one has nothing to do with energy compounds or compounds that are used for energy production or source of energy. But rather, this one is degradation leading to formation of the compounds that are nitrogen-containing compounds, just like we mentioned in the class. So we will see here 7 amino acids that are degraded to acetyl-CoA. One of them is tryptophan, and we have seen how tryptophan is being, a part of tryptophan is being converted to or clipped to remove alanine in positive reaction. We will see what happened in these next reactions and how does that lead to formation of acetyl-CoA and finally lead to acetyl-CoA. And we'll also see lysine, phenylalanine, tyrosine, leucine, isoleucine, and threonine. For the threonine, we've already seen, because threonine completely degrades to produce what? It produces acetyl-CoA and a glycine. And the glycine, we've seen how the glycine is converted to serine, and serine converted to adenine. So we don't need to talk about threonine degradation. We'll see how tryptophan, or the remaining component of tryptophan, is being converted down to the lysine also. So let's go. Remember, we talked about some amino acids that are exclusively ketogenic, like lysine and leucine. So we will see from this degradation pathway where they are exclusively ketogenic. They don't produce anything that can maybe use later for the synthesis of glucose. Now, we have already seen how tryptophan through post-step reaction lead to formation of alanine. Now, through 9-step reaction, remember I said I don't want to give the breakdown because it's a very long and cumbersome reactions involving breakage or degradation of tryptophan. So through 9-step reaction here, we have the alanine already been released since from the 4th step, now including the 2 carboxyl group in form of carbon dioxide to form what we call alpha keto adipose. So the same alpha keto adipose is also produced by the breakdown of lysine. The breakdown of lysine, including the removal of the amino group, the transamination reactions, and there is oxidative decarboxylation where, and there is oxidation reaction where oxidation takes place. So first of all, we have the transamination, the removal of this. And we know in transamination reaction, after the removal, the oxidation takes place first, and then the transamination reaction. So you have the transamination reaction. The transamination reaction introduces the carbonyl group, and then finally leads to formation of this alpha keto adipose. The alpha keto adipose undergo oxidative decarboxylation reaction. The oxidative decarboxylation reaction involves the oxidation of alpha keto adipose and then removal of carboxyl group. At the same time, addition of coenzyme E from what we call glutaryl CoA. So glutaryl CoA2 also post-step reaction release a carboxyl group to form acetoacetyl CoA. Now we have seen how tryptophan, the indole ring of tryptophan, is being broken down to form acetoacetyl CoA through about 9, 10, or 10-step reaction. While in the case of lysine, we have 9-step reaction leading to formation of acetoacetyl CoA. And which we said acetoacetyl CoA can easily be converted by ketoacyl CoA transferase, which will get coenzyme E to the 1-acetyl CoA of the acetoacetyl CoA, because when you look at it, you can see this one is acetyl CoA on its own, but this one, when you attach a coenzyme E like this one to it, it would also form another acetyl CoA. So you can have 2 acetyl CoA from this acetoacetyl CoA. Now, from the other hand, we have phenylalanine oxidized to form tyrosine. In the reaction, we've already given an example, although we'll talk about this reaction in detail because of some genetic defect involved in the enzymes involved in the metabolism of this phenylalanine and tyrosine. So the next reaction, we'll see the breakdown of how this reaction takes place because of the interest in this genetic defect. So we have phenylalanine oxidized to tyrosine, and then tyrosine through pipe step reaction leading to formation of fumarate and acetoacetate, which I told you, see how this fumarate and acetoacetate comes from. The acetoacetate can now combine with coenzyme E to form acetoacetyl CoA. In most cases, now you have an succinyl CoA responsible in the transfer of this coenzyme to form succinate, and then acetoacetate is converted to acetoacetyl CoA. Now, from the other hand, also we have another branch chain fatty acid called lysine, which we said is also exclusively. Now we can see lysine form acetoacetyl CoA and nothing else. So acetoacetyl CoA lead to formation of acetyl CoA. And then acetyl CoA can only use for energy production, or it can be used for fatty acid synthesis, or it can be used for ketone bodies. So it is exclusively ketogenic, cannot be used to produce glucose. And then another one, which is leucine. Leucine is broken down which this reaction involves transamination, decarboxylation, and carboxylation reaction, including 6-step reaction to produce acetoacetate. This acetoacetate, like the earlier one, you said it can undergo the transfer of coenzyme E from succinyl-CoA to form acetoacetyl-CoA, while the other parts form acetyl-CoA. Now, we have also isoleucine. Isoleucine can also be converted to propenyl-CoA and acetyl-CoA. In the reaction, we talked about conversion of propenyl-CoA by propenyl-CoA carboxylase to dimethylmalonyl-CoA. We say a 3-step reaction to succinyl-CoA. So we can see the step involving the formation of dimethylmalonyl-CoA one step. And the second step, L-methylmalonyl-CoA, second step. And the third step is the L-methylmalonyl-CoA to succinyl-CoA. We'll also see the reaction. So this is basically how all these 7 amino acids were converted to acetyl-CoA. But remember, this isoleucine, in addition to acetyl-CoA, it also produces propenyl-CoA, and the propenyl-CoA will be used to produce succinyl-CoA. In addition to that also, phenylalanine and tyrosine together produce fumarate and then acetoacetate. So acetoacetate is the one that is involved in the formation of acetoacetyl-CoA and then finally to acetyl-CoA, while fumarate can be used for the synthesis of glucose. So these enzymes are still glucogenic, phenylalanine and tyrosine. So, breakdown of phenylalanine and tyrosine, the same pathway. So we've already talked about how phenylalanine became oxidized to form tyrosine. The reaction we said catalyzed by phenylalanine hydroxylase. And we talked about this enzyme being a mixed function oxidase, which uses a molecular oxygen to produce water. And we said classically this oxidation reaction involved tetrahydrobiopterin, where we say dihydrobiopterin reduced or became reduced by an enzyme dihydrobiopterin reductase, and then to form tetrahydrobiopterin. Then tetrahydrobiopterin is responsible now in reducing the water molecule to form, oxygen to form a water molecule, and then the other oxygen is being attached to phenylalanine to form tyrosine. So once tyrosine is formed, you have the next reaction will continue. But this phenylalanine hydroxylase, there is a genetic defect that is being reported. There are some individuals that have an ability to convert phenylalanine to tyrosine because the enzyme have a defect, have a problem that cannot convert phenylalanine to tyrosine. So when you take a food that contains a lot of phenylalanine, or when there is a lot of phenylalanine in your system, it need to be degraded, need to be broken down. This phenylalanine will accumulate. And the accumulation of phenylalanine lead to a condition we call phenylketonuria. Why is it called phenylketonuria? Because phenylalanine will find another pathway for degradation. And that pathway involves, number one, transamination of phenylalanine. And once phenylalanine becomes transaminated, where the amino group is removed, once the amino acid is grouped here, we have what we call phenyl pyruvate. So this will become pyruvate. This is the phenyl group. We have phenyl pyruvate. So accumulation of this phenyl pyruvate, because phenyl pyruvate is a keto group, that is why it is called phenylketonuria. So you have accumulation of phenylalanine and the phenyl pyruvate. The phenyl pyruvate also can be decarboxylated, where a carboxyl group is removed. And once the carboxyl group is removed, it forms what we call phenyl acetate. Phenyl acetate. Or it can be reduced So you have, what am I saying? Phenyl acetates, or the phenyl pyruvate can be reduced to form phenyl lactate. So in this phenylketonuria, due to accumulation of phenyl alanine can be converted to so many. Can be converted to phenyl pyruvate to phenyl acetate, or phenyl lactate. So in an individual that cannot convert phenyl alanine to tyrosine, you have an accumulation of all these substances, including the phenyl alanine step, the phenyl pyruvate, the phenyl acetate, and the phenyl lactate. So this compound accumulate and they can be excreted in urine. And of course, the smell or the odor produced by the urine of those individuals has been attributed to the phenyl acetate due to the presence of phenyl acetate. And the individuals with accumulation of these things were characterized to have defective neural development. Because remember, we talked about some of these amino acids are neurotransmitters. If they are not neurotransmitters, some of them are precursors of neurotransmitters. If they are not precursors of neurotransmitters, some of them are antagonists of neurotransmitters. So they are, to some extent or to a high extent, being related to neurotransmission. So problem in their metabolism can affect neurotransmission. So from the neural development to the activity of the neuronal cells. So therefore, if you have any deficiency in the metabolism, you will see that the deficiency in the metabolism can somehow be related to the defective neural development and some mental retardation. That's aspect of mental retardation. So this phenylketonuria is being characterized by defective neural development and mental retardation. So now let's move ahead. We have a tyrosine produced in a situation where phenylalanine hydroxylase is not defective. So you have tyrosine. Tyrosine can be deaminated by an enzyme transaminated by enzyme transaminase or aminotransferase. Remember, I mentioned aminotransferase and transaminases are the same enzymes. So you shouldn't be confused, please. They are the same enzymes. So you have this alpha ketoglutarate now accepting the amino group to form glutamate, and you have para-hydroxyphenyl pyruvate. So there are also a situation where an individual will have defective tyrosine aminotransferase. This leads to accumulation of tyrosine. And tyrosine accumulation due to defective tyrosine aminotransferase, we call the condition tyrosinemia 2, because there are a number of tyrosinemia. So this one is called tyrosinemia 2, and it is characterized by accumulation of tyrosine and also phenylalanine and also phenyl pyruvate and all the other one, phenyl acetate, phenyl lactate, a little accumulation of this. And this is also related to the problem of the mental retardation. Remember, they are all related to neurotransmission. So like I said, it has to do, it's all related to mental retardation and specifically also related to pain in the eye, production of tears, the rest. And one of the mechanisms for the treatment is to ensure that the diet do not contain as much as phenylalanine to reduce the effect of this. Then the next is in a situation where tyrosine aminotransferase is functional, you have this para-hydroxyphenyl pyruvate converted to homogenticate. Homogenticate by an action of para-hydroxyphenyl pyruvate dioxygenase, which are simply oxidation reaction. Dioxygenase is also a form of this oxygenase, but this one is dioxygenase different from the monooxygenase. So Also, deficiency in this dioxygenase leads to tyrosinemia, and this is also classified with accumulation of this hydroxyphenyl pyruvate is also related to the mental retardation, the problem with coordination. That individual have problem with coordination, which all has to do with the problem of neural development, as we earlier mentioned. So the next reaction is conversion of homogenticate to mallyl acetoacetate. So mallyl acetoacetate is just like, okay, you don't know this thing that happened. It's just a mallet, which is what is what we call it, an intermediate of citric acid cycle attached to acetoacetate. So this enzyme, homogenticate 1,2-dioxygenase, does the work. So the absence or genetic defect in this enzyme leads to a condition called alkaptonuria. And this alkaptonuria is due to accumulation of homogenticate. And homogenticate can be excreted in the urine. So excretion of homogenticate in the urine, and this homogenticate, once it's released out from the urine, it can easily oxidize in the presence of oxygen. So oxidation of homogenticate leads to formation of compounds, and the compound that is newly formed or oxidized homogenticate can polymerize to form a polymer called alkapton. So this alkapton give a black color to the urine, so urine will turn black. That is why it is even called black urine disease, alkaptonuria. So in a situation where homogenticate is homogenticate 1,2-dioxygenase is functional, is not deformed, then this mallyl acetoacetate is converted by an isomerase to fumaryl acetoacetate. This is just an isomerization reaction where the cis is converted to trans. So we have fumaryl acetoacetate. The fumaryl acetoacetate by an enzyme fumaryl acetoacetate, which is more or less a hydrolase, will now break the fumaryl acetoacetate to fumarate and acetoacetate. Now we've seen how phenylalanine and tyrosine are converted to either fumarate or acetoacetate. And we've already talked about acetoacetate being converted to acetoacetyl-CoA in the presence of succinyl-CoA, which leads to formation of succinate by 3-keto acyl-CoA transferase. So this is basically reaction involved in phenylalanine and tyrosine catabolism and leading to formation of all these disorders that are attributed to their catabolism, which we will talk about. I will give just a summary, so I don't need to talk much about them since already done here. So, and this one, I think I've already discussed about it. Genetic defect in phenylalanine catabolism leading to formation of phenylketonuria, and we talked about why phenylketonuria, how is the phenylketonuria all about, and the alternate pathway of phenylketonuria leading to formation of phenyl pyruvate, and phenyl pyruvate is converted to phenyl acetate and phenyl lactate. The only thing I didn't mention is that transamination of phenylalanine involves pyruvate, where alanine is produced in the amino phenylalanine amino transferase. So you have phenyl pyruvate. Phenyl pyruvate, we said, which is decarboxylated to form phenyl acetate, which gives the odor of the urine in individual with phenylketonuria. And we also have phenyl lactate, which all form the component that is being secreted in this condition. We also talked about alkaptonuria resulting from the accumulation of homogenticate. And we said one of the characteristics is that individually, these alkaptonuria are prone to develop arthritis. Okay, so our next reaction, as our next class will start with the 5 amino acids converted to alpha-ketoglutarate. So please, I will advise you to your time, please, and read this. I will try and see when I can, so that I will just give a summary. See how I'm taking my time to go through this thing and talk about each reaction and what happened in each reaction. I want to be sure that if you actually go through this thing, you will not have a problem, or you will not have much to even ask. But nevertheless, we'll still revive this when we come to class, then ask questions in areas you don't understand, so that we'll continue from where we stopped. I wish you all the best.